right, hello. My name is Rachel Baines, and I'm from Evangel University. And today I'm presenting on the crystal structure of dihydrolipoyllysine-Residue Succinyltransferase, component of 2-oxoglutarate dehydrogenase complex. That's a mouthful. So I have a story for you. We originally tried to crystallize this one protein, and then along the way ended up crystallizing another protein. So, HAT is a human airway trypsin-like protease. It has some biological functions. It mainly plays a role of inserting viruses into human cells, and this mainly occurs in influenza as well as the SARS-CoV virus. It's also involved in skin cancer, and currently the three-dimensional structure of HAT is unknown. So our first aim was to express and purify HAT, and then go on to find the crystal structure. So first starting out for our research was to insert the plasmid containing HAT into an E. coli cell. We use BL21 cells to do this. We heat shock the cells, which basically breaks open the bacterial wall, creating little holes, and this allows the bacteria to accept the plasmid as its own DNA. And then we put these on agar plates, LB agar plates, and started expressing them at 37 degrees Celsius overnight. And then we were able to extract single colonies from these plates to start starter cultures. And then once these starter cultures started growing, we used IPTG to introduce the activation of protein. So once the bacteria was able to accept our DNA, IPTG then turned on the protein to start making, or just turned on the bacteria to start making our protein. So once we have a bunch of protein within our E. coli cells, we then want to be able to purify this. So we do this by sulfication, which is high-frequency sound waves that breaks open the bacterial wall, and so then you have all your cell components floating around in solution. So this is cell membranes, organelles, proteins, everything in the cytosol. So in order to separate this out, we centrifuge it right here, and so this creates basically two main phases. We have a solid phase, which is most of the membrane and the organelles, and then a liquid phase, which we call the supernate, and this is a lot of the smaller components that are within the cytosol. So this includes our protein, as well as other small molecules. So then we take the supernate, which I said is the liquid portion, and we want to further purify this. And we do this by affinity chromatography, and this isolates this his tag on our HAT gene. So when we went to put our HAT gene in the plasmid, we added a 6-histidine tag, and this has a high affinity for cobalt. So these cobalt beads are within the column, as you can see right here. And so when we pour our solution in, the his tag will then bind to these beads and hold on to our protein, and everything else will then drain through. We will do 7 rinses, which rinses out other molecules that are attached to the beads, weakly associated, to then just get our protein attached to those beads. Now finally, to extract that protein, we use imidazole, which displaces the bonds on the cobalt beads. So it releases the bonds between the histidine tag to the cobalt beads, and it leaves us with our protein in this solution. So there, ideally, we have a solution with just purely our protein. So from here, just to check that we have it purified, we run it on an STS-based gel. And so basically what this gel does is it separates out proteins by molecular weight. And so the ones that have a higher molecular weight will get stuck towards the top of the gel, and the ones that are low will make it to the bottom. And we are looking for our protein at 47 kilodaltons. So that is right about here. So we have our standard ladder, and this is all the known molecular weights of these proteins. So the ones we're focusing in on is this one right here, which is at 50 kilodaltons. So next we have our protein pellet, and then our lysate, and then our column flow through. If we're thinking back to the column when we're pouring our protein through, all the liquid that comes off initially is what is in this column right here. The next two are rinses, so these are just basic rinses that are going to help wash through any proteins that are kind of stuck on the beads. And then the 7 and 8 are washes, which will loosen any weakly associated proteins that are bound to the beads. So then the last one will be your elution. And in an ideal world, there should be nothing in this column except for a tiny little band of your protein. But as you can see here, we do have several other protein bands. I know it's kind of faint, but we have some significant ones here and here. However, we did, after this expression, we did find that this was pure enough, so we went and tried to grow some crystals out of it. So in order to grow crystals, you have a bunch of liquid with your protein in it, and you need to concentrate that because it's too dilute right now. So what I'm going to do is centrifuge this, and so this spins off a bunch of extra liquid, like I said, concentrating your protein. We then used commercial screens to identify or to set up this crystal structure using a hanging drop method. So you basically have a plate and it has a bunch of wells in it, and then you use each one of these crystal screens and put 750 microliters within the well. So that's captured right here. Then you take a cover slip and you're going to put 1 microliter of the solution on it, as well as 1 microliter of your concentrated protein, and then seal it. And so we used immersion oil to seal it. And this creates pretty much a perfect environment for growing crystals. So you just want the proper crystal structure, or the proper conditions will then grow these crystals. So we did get crystal heads. We had several different crystal heads, but the one that gave the most prominent crystals was this one right here. It was 0.2 molars of calcium chloride dehydrate, 0.1 molar of Hb sodium, and 28% of pig 400 at about a pH of 7.5. And so these are our crystals, and they're pretty large in size. We're actually very surprised with this yield. So now to collect these crystals, we have these really tiny loops, they're little microscopic loops, and we grab the crystal with it and then freeze it in liquid nitrogen. And then we send these crystals over to SLAC. So SLAC is the National Accelerator Laboratory, and I believe it's 1 out of 17 laboratories that's run by the U.S. Department of Energy, and it's operated at Stanford University. So this was a really cool experience. This lab is located in California, and we were able to control our crystals going into the beam from Missouri. So we were able to watch this online and actually get to do it, which was pretty awesome. Our crystals were located in this linear beam. This is where our accelerator was, and so that was really cool. And I also want to acknowledge Dr. Emily Scott and Dr. Audrey Lamb, who gave us beam time and able to do this. So now we have the data collected from our X-ray radiation of the synchrotron accelerator, and we put this data into IMOS film to process the data. And we found out that our crystal structure was a 6-point angstrom structure. Now this is really, really great. Usually structures below 3 angstroms are publishable. So we were really excited. We're like, yes, we have the structure of HAT. It looks great. So now let's go publish it. So we did molecular replacement and with HAT, and we found out it failed. So this basically means that the protein that we crystallized wasn't actually HAT. And at this point, we had no idea what it was. So like, okay, we have this really, really good crystal structure, 2.6 angstroms. Now we need to figure out what we crystallized. So we decided to send off samples of our crystals for mass spec at MS Bioworks. And then we received a list of 40 different proteins that were either in the crystal or in the solvent that we sent it in. And so we went through down the line doing molecular replacement, which with each of these, not the ideal way to do crystallography, but we really wanted to know what was in this structure. And so we came to this one, which is SUCB, and this gave us the correct mathematical solution for our protein structure. And we were able to get the electron density from that. And I'll show you that picture in a minute, it's super cool. But now to find out what even is SUCB, we didn't know. So we did some research, and SUCB is an enzyme in E. coli, and it's involved in multiple biological processes. But the main one to hone in on is the Krebs cycle, or the citric acid cycle. And so we can see that here. We all know that cycle, but if you don't know it, you'll know it in biochem. But we found out that Suc-B catalyzes the overall conversion of 2-oxoglutarate to succinyl-CoA. And if you look right here, that's this product. So think of this as a side reaction that basically produces a product going into the Krebs cycle. So pretty cool. And we also found that it aids in the degradation of amino acid L-lysine. So now going on to show you our diffraction data. So like I said, we saw the structures, 2-point angstrom structure. So this is a really high resolution, high quality structure. And we found that multiple complexes of Suc-B actually form to make up the core of this structure. So 24 in total. And our crystal structure was a higher resolution than has been previously published in literature. So that was pretty cool too. And I want to explain this picture right here. So this is the density. We have in the blue, this is the electron density that we got from the data at SLAC. And then the green bars, these are the amino acids of Suc-B that we actually modeled into the density. And so by doing this, you can get the full structure of Suc-B. So now looking at the tertiary structure. So the picture I just showed you, from here, from collecting this data, we can put into another program, and that gives us this tertiary structure. We see parallel and anti-parallel beta sheets, which are located here. Here's your parallel sheets, here's your anti-parallel sheets, and then also 5 helical structures that kind of wrap around into a 2-layer open phase motif. So another interesting thing is that Suc-B, like I said, so there's 24 of them that form this overall multi-enzyme complex. But Suc-B forms a trimeric unit. So this means that it has to form with, or one monomer forms or binds with 2 others, and then that's when it becomes biologically active. So more commonly, these enzymes, with the one monomer they're active, but this actually it takes 3 to make them biologically active. And so the catalytic site forms the core of this 2-oxoacid dehydrogenase complex, and the interactions are stabilized by hydrogen bonds as well as salt bridges. So these are kind of weakly associated bonds. So now looking at the catalytic site, if we're going to kind of look in from this angle here, which is this picture. So I'm kind of going to help you orient. This green helix is located right here, and this blue helix is located right here. So we're looking at the catalytic domain where the Succinyltransferase activity occurs. And so basically this catalyzes the overall conversion of 2-oxoglutarate to succinyl-CoA and carbon dioxide, feeding into the Krebs cycle. So our conclusions, we have a 2.6 angstrom structure. So this is a big deal for us. Our further directions, we're going to continue refining our Suc-B protein model, and we're going to use different methods to obtain a crystal substance like within the protein structure. And we plan on submitting our structure for publication. And then some acknowledgements. I would like to thank my amazing team. We have Dr. Matthew DeBoer, who was the PI who started this project, who got it up and running. And then you have me, you have my partner Joel Manzi. He actually graduated, but he first started this project with me. And then my amazing partner Tricia, he actually did a lot of the research that went into this. And then Dr. Natasha DeBoer, which is incredible. She helped us through a lot of this project, mentoring us and helping us with next steps. And some collaborators I would like to point out is Dr. Emily Scott and Dr. Audrey Lamb, who not only donated supplies to make this project possible, but also donated me time. Thank you once again.
All right, can you talk to your hydrogen crystallization?
I'm so sorry. Maybe. Alright. Very good. This one you talking about?
Yeah, what's the driving force for the formation of the crystals here? A lot of scientists, they show crystal aggregates, things like the introduction of ionic solvent. So what's driving the formation of the crystals?
So basically, I'm sorry. So basically, this is the important factor here is it's creating an environment to form these crystals. So we're using commercial crystal screens that have been shown in the past to actually produce crystals, and so we use a variety of them, and the crystal is mixed with the solution, and then it's also here. So as it evaporates up, it creates this environment that eventually forms the crystal. Yes, the crystals will form right in here in this little dot. And so later, our crystals form between 2 days and a week-ish. And so we'll look at this under a microscope and you'll see the crystals are forming within here. And you'll get some precipitate in some of the wells, but the fine crystals are just solely crystals and not so much precipitate.
So what's the head and miss ratio like on this? Because I know from the early days of protein crystallography, people would have hundreds if not thousands of these set out in various rooms.
Right.
So how often do you see crystals?
How often do we see crystals?
Yeah.
These ones actually showed up in a lot of wells. I think we got, my PS could correct me on this, but 4 good hits with fairly large crystals, but we saw crystals in a fair amount of the wells. Like I said, we're trying to crystallize hot, so we try to use some of the conditions that could crystallize that. So
I'm very sold on the idea of the triner. Sometimes you see artifacts of crystallization, things will come together when you're forcing them to grow crystals. Is there any evidence for your 24 monomer state actually in the natural system, not in the crystals? So sometimes when you form crystals, you force things into weird configurations, you bring things together that might not be kind of found together. Is there any evidence out there looking at this in its natural habitat of this 24 monomer state being out there?
gosh, I think I understand your question. So the 24 monomers forming this overall complex has actually been published in previous literature. And that's how we found that it was 24 monomers. It's really cool. I wish I had a picture of it, but it forms a cube-like structure. So 2 of these trimeric units kind of like form on top of each other, and they form a corner of the cube, if that makes sense. So you'll have 6 units in each of the corner. I don't know if
that's what leaves your active sites open.
So yeah, it actually does. So this right here, if you could try to imagine a donut with me, the donut will go around this way, and so the center of this multi-complex enzyme has like a hole in it, and so this is one of the active sites. As you can see here, we looked at the active site going in from here. I don't understand the whole kinetics behind it. We haven't done that much research on it, but yes, it does allow for the active sites to be open to substrates.
So, really great presentation. You talked about how you've got clear resolution image. I'm just interested, have you found any new or different structures in your different literature?
Have we found any new structures of
So the structure of this protein, is it the exact same as the previous literature, or have you found any new setup? Is the structure that you have found different than previous literature?
What was the resolution of the previously published version?
Right, I understand. I don't know the exact resolution, but when we're talking about resolution of crystal structures, we're talking about this angstroms. And so that's basically showing the clarity of amino acids that...
I think what my colleague is asking you is
Yes.
you go from the previous resolution to your 2.6 angstrom resolution. Are you bringing out any new details that previously were in C?
So this is more of a clearly proven structure. Like I said, also with our research, we're going to try to model in a substrate with our complex. And so this will also give us high chances of publication. But the previous published structure was quite a while ago, and so this just brings more clarity to the structure. Is there any other question?
Was that second band or you were
Come back. Here. Yes, we think so. I believe it actually is this one up here, I believe. clear to tell. This is a bad gel picture, but you do have a lot of protein in here. So it would be kind of hard to point out where it exactly is. Yes.
So, how do you get this protein? Did which tag
Histidine tag?
Did that totally attach to this protein?
Oh, yes. Okay, so I didn't talk a lot about the gene and putting that into the plasmid. But when we created our plasmid, we did two things to the hat gene. We made it ampicillin resistant, and then we added a 6-histidine tag. And so this tag has a high affinity to cobalt. So ideally, when your bacteria makes your protein, at the end of your protein, it's going to have all these histidine amino acids, right? And that binds to it. But no, that wouldn't be on the Suc-B. So basically, Suc-B has some affinity to cobalt. And it also could be that we just produced so much of it because we were trying to purify half. And over our different processes, we weren't getting the best results. So we just grew a ton of E. coli, and we think that it was either it has affinity to cobalt or it was just so much of this protein. That's what we ended up crystallizing.
So it wasn't necessarily the tag?
No, the tag.
Itself,
Correct. The tag, yes, sorry, the tag is definitely was on the hat protein. It does not go into the Suc-B. Awesome. Any other questions? Yes.
So how does the crystal structure of this protein, like earlier elucidate the function of it in a real biological setting? The crystallized structure and looking at it, like they're different. The composition changes. So how are you able to account for? We know that this will, like, it'll have the same effect in like just anything, like real biological setting. What's the difference between that and like the liquid nitrogen frozen crystal structure?
Right. So when you're crystallizing proteins, this is kind of like the preliminary stuff. You're just basically finding out the three-dimensional structure of it. You can't know kinetics of it because it's already, you can think of it as it's already frozen in place in one conformation. So how we know that it does all of that and it feeds into the citric acid cycle is from previous literature that we've read up on. So if you would want to do more, or you would want to do more about figuring out how the active site works and how that's all biologically active, we would have to do kinetic studies, which are a little bit different than this. Does that answer your question?